Welcome to the Sans Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Please like and subscribe. It really helps support the Sans Science Experience and allows us to keep bringing you these deep dives. Imagine a tiny, ancient genetic blueprint passed down solely from your mother that might hold the secret to how long and how well you live. Today, we're embarking on a deep dive into precisely that. The fascinating and complex role of mitochondrial DNA, or mtDNA, haplotypes in determining lifespan. Think of this as your shortcut to understanding the very edge of aging research.
Yeah, it's a really exciting area. We'll be exploring how this inherited mitochondrial variation isn't just, sitting there passively. It actively influences aging. It does this through profound effects on our cells' energy metabolism, on the production of reactive oxygen species, ROS, and through this incredibly intricate dance between the mitochondrial genes and the nuclear genes. And while the basic mechanisms seem pretty conserved across species, what's really captivating is how, well, context-specific the effects often are.
So, what does this all mean for how long we live? I mean, right down from tiny worms all the way up to us.
Okay, let's unpack this a bit. To really get how these cellular powerhouses might dictate lifespan, we need the basics first. What are mitochondrial genomes? I gather they're quite different from our main nuclear DNA.
They are very different. Think of it like this. Instead of the huge library in the nucleus, you have this small, separate circle of DNA right inside the mitochondria. It's inherited just from your mother, which is key. And it doesn't mix and match, recombine like nuclear DNA does. Instead, you get these distinct patterns, variations. We call them haplotypes. These haplotypes have subtle differences in the genes that code for proteins, for RNA molecules, and even in the regions that regulate how active those genes are.
Okay, so different patterns passed down maternally. But why do these subtle differences matter so much?
Well, that's the crucial point. These variations aren't just like neutral markers. They're not just labels. They directly impact how well oxidative phosphorylation, or OXBHOS, works.
That's the cell's power plant, right? It's exactly the core energy production system. And these variations also change how much ROS, reactive oxygen species, gets produced. You can think of ROS as sort of the exhaust from that power plant. A little is normal, maybe even useful, but too much causes damage. So these small genetic differences in your mtDNA, they directly tweak an organism's basic physiology and, ultimately, its lifespan.
It sounds like this isn't a brand new idea then. Scientists have been looking into this for a while.
Oh, absolutely. Decades, really. Research over the past 20, 30 years has shown this influence again and again across lots of different model systems: worms, flies, mice.
And finding it consistently across different life forms must have been pretty compelling.
It really has. It reinforces that we're likely looking at a fundamental biological principle here, not just some quirky thing happening in one specific species.
Okay, so we have the basics. Let's see it in action. Where better to start than the workhorse of aging research, the tiny worm, C. elegans? What have these little guys taught us about mtDNA and how long we live?
C. elegans provides some really clear examples. In these worms, we see a very direct link between their mtDNA haplotype variation and how efficient their electron transport chain is. That's the core machinery for energy. For instance, there's a specific known difference, a single letter change, technically a non-synonymous substitution, in a gene called COX1. This gene makes a part of complex 4. Now, this isn't just a tiny tweak with no consequence. It actually affects how well that enzyme works and changes the amount of oxidants like ROS produced.
And the impact on lifespan.
It's significant.
Yeah.
In one common lab strain, CB4856, this single nucleotide difference correlates with about a 25% shorter lifespan compared to the standard N2 strain.
25%. Just from one letter change in the mitochondrial DNA. That's huge.
It really is.
Yeah.
And it points toward a really key finding, something that makes you rethink things a bit. We know that damage to the mtDNA does build up as organisms get older. That definitely happens. But what seems more decisive, perhaps, is the inherited mtDNA haplotype, the sort of innate quality of the mitochondrial genome you start life with. It seems to set a baseline for energy metabolism, for ROS generation, essentially laying down the track for an organism's life history trajectory right from the start.
Wow. So it's not just about the wear and tear that happens over life. It's more about the quality of the original mitochondrial operating system you inherit.
That's a great way to put it. It fundamentally shifts the focus. It's not just about damage accumulation and repair, but also about the inherent quality of the design you begin with.
That makes sense. Like the foundation of a building versus minor cracks later on.
Precisely. The blueprint you inherit seems to, pre-program a significant chunk of your potential lifespan, at least in these models.
Okay, worms show a pretty direct link. But you mentioned things get more complex. Let's move up the ladder to fruit flies, Drosophila. You said this is where it gets really interesting.
Yeah, Drosophila research has given us some incredibly detailed evidence, especially on the interactions. First off, they've clearly documented lifespan differences. There are classic experiments where they take different mitochondrial genomes, say from fly populations originally from Dahomey in Africa or Japan or Alstonville in Australia, and they put these different mtDNA types into flies that have the exact same nuclear genetic background, standardized nuclei.
Controlling for the main genome, basically.
Exactly, and the results are pretty stark. Compared to the Alstonville line, which they often use as a reference, flies with the Dahomey mtDNA lived about 4% shorter on average. But those with the Japan haplotype, their lifespan was reduced by up to 15%.
15% again. And is the reason the same as in worms? Energy and ROS?
Fundamentally, yes. It boils down to differences in OGXBHOS efficiency and how much ROS is produced. The proteins encoded by the mtDNA variations can change how the OGXBHOS complexes those multi-protein machines assemble or how well they function. This affects the ATP output, the energy currency, and the amount of ROS generated as a byproduct. We see this in related fly species too, like Drosophila simulans. Different mtDNA haplogroups are linked to different survival rates, seemingly tied to variations in ROS production and the fly's antioxidant defenses.
Okay, but you mentioned interactions. This sounds like the mtDNA acting on its own. Where do the interactions come in?
Right. This is where it gets really critical. Remember, most of the proteins needed for that cellular power plant for OXPHOS are actually made using instructions from the nuclear DNA, not the mitochondrial DNA.
Oh, right. So they have to work together.
Exactly. They have to be compatible. The interplay between the mtDNA variations and the nuclear background is absolutely paramount. Scientists use clever experiments called introgression studies to show this. They carefully place mtDNA from one genetic line into a different nuclear background. And these studies clearly demonstrate that these interactions can either sort of cancel out negative effects of mtDNA, or they can make them much worse. This is what we call mitonuclear epistasis.
Epistasis. Okay, break that down.
It just means the effect of one gene, in this case a mitochondrial one, depends on the version of another gene, usually a nuclear one it interacts with. Think of it like two musicians. The effect of what one plays depends entirely on what the other is playing, harmony or discord.
Got it. That makes perfect sense. You also mentioned something really intriguing about sex differences, the mother's curse.
Yes, that's a fascinating wrinkle. In some fly studies, certain mtDNA haplotypes seem to have a much bigger negative impact on the lifespan of males compared to females.
What would that be?
Well, think about how mtDNA is inherited only through the mother. So natural selection really only sees the effect of the mitochondria in females. If a mutation arises in the mtDNA that harms males but doesn't really affect female reproduction or survival, selection might not weed it out effectively. It can persist across generations simply because it's passed on by the mothers, even if it shortens the lives of their sons.
Wow. So evolution is essentially blind to male-specific harm in mitochondria? That feels like a built-in vulnerability.
It kind of is. It highlights a really interesting evolutionary dynamic. And we see echoes of this in disease models too. For example, in fly models of Leigh syndrome, a severe neurodegenerative disease often caused by nuclear gene defects affecting mitochondria, the specific mtDNA haplotype the fly has can significantly change how severe the disease is and how much lifespan is reduced. It's another powerful demonstration of that mitonuclear pistis, the mitochondrial background, modulating the effect of a primary nuclear problem.
Okay, worms, flies. The principles seem consistent but gain complexity. Let's move closer to home. Mice. What do we learn from mammalian models?
In mice, we definitely see these same core ideas playing out, but maybe with even more intricate regulation. One tool researchers use are Cybrid models. Cybrid stands for cytoplasmic hybrid. They essentially take cells, keep the nucleus the same, but swap out the cytoplasm, including the mitochondria. So you have the same nuclear genome paired with different mitochondrial genomes. And these studies show quite clearly that the mtDNA haplotype influences how well cells grow and function, especially when they really need efficient OXPHOS.
So cellular evidence is strong. What about lifespan in the whole animal?
We have direct evidence there too. Studies using mice engineered to have defects in mitochondrial DNA replication. The Polkin mutant mice, they're often called, are very informative. Even starting with low levels of inherited mtDNA damage or specific variants in these mice can lead to signs of premature aging and a shorter lifespan. It really emphasizes that the quality of the mitochondrial genome you inherit from your mother is a crucial factor. And again, the interplay between those mtDNA features and the mouse's nuclear genetic background is key in driving those aging phenotypes.
But you also mentioned other factors coming into play in mice. Environment, compensation.
Absolutely crucial point.
Yeah.
In mammals, the effects aren't always straightforward because the body has ways to respond and adapt. For instance, maybe a particular mtDNA variant slightly increases ROS production. That sounds bad.
Yeah.
But the cell might respond by boosting its antioxidant defenses or by making more mitochondria overall. That's called mitochondrial biogenesis. These compensatory mechanisms can sometimes buffer or mitigate the potentially negative effects. It's a much more dynamic balance than perhaps in simpler organisms.
Okay, so it's the inherited blueprint, the interactions with the nucleus, and the body's ability to compensate.
Exactly. It's a complex system.
Which brings us, inevitably, to humans. What does all this mean for us? How does our own mitochondrial DNA influence our health and how long we live?
Well, in humans, researchers mostly rely on large association studies, looking for links between specific mtDNA haplogroups, those ancient lineages, and longevity or age-related diseases. And there have been some interesting findings. For example, haplogroup J, which is common in Europe, has been linked in some studies to increased longevity.
And do we know why?
The thinking is that haplogroup J might confer a mitochondrial profile that's slightly less efficient at making ATP, but crucially produces less damaging ROS as a result, a kind of trade-off that potentially minimizes long-term oxidative damage. There are also specific variations, polymorphisms, like one called MT5178A in the NT2 gene, that have been linked to lower risk of things like atherosclerosis. And obviously, avoiding cardiovascular disease helps with longevity.
So some of us might genuinely inherit mitochondria that give us a bit of an edge for a longer, healthier life.
It seems plausible, yes. But, and this is a big but, these associations are often very context-specific. What holds true in one population, say Northern Europeans, might not be found in another population with a different overall genetic background. Sex also matters, as does environment, diet, lifestyle, exposure to toxins.
So no universal longevity gene in the mitochondria.
Not a simple one, no. It's more nuanced. This leads to an interesting idea called the genetic golden mean hypothesis. The suggestion is that mitochondrial genomes that are closer to the long-term evolutionary average, meaning they haven't accumulated too many potentially detrimental variations, might function more optimally and be associated with greater longevity. And there's some evidence supporting this. Studies looking at centenarians, people living past 100, sometimes find they tend to have fewer amino acid changes in key mitochondrial proteins compared to people who develop age-related diseases earlier.
Like they've managed to preserve a more consensus mitochondrial design.
Kind of like that, yes. A sort of evolutionary sweet spot. And of course, mitonuclear interaction is just as critical in humans as in flies or mice. That compatibility between our two genomes is vital for health and likely influences susceptibility to many age-related conditions and ultimately lifespan.
But you mentioned controversies. It's not all settled science.
Definitely not settled. It's important to be clear about that. Some very large studies looking at huge numbers of people haven't found consistent, strong links between specific haplogroups and living to extreme old age.
Why the discrepancy?
It could be several reasons. Maybe the effects are quite subtle and hard to detect statistically without enormous, perfectly matched groups. Or maybe underlying differences in the populations being studied, what scientists call population stratification, are muddying the waters. It really highlights that we need more research. We need better tools. High-throughput sequencing, looking at huge, diverse populations, combining genetic data with data on proteins and metabolites, what we call multi-omics. That's likely the way forward to really untangle this complex puzzle in humans.
It's been quite the journey. Worms, flies, mice, humans. If you had to boil it down, what are the big overarching themes? The grand synthesis.
I think there are a few key takeaways that echo across all these different species. First, the fundamental mechanism is conserved. mtDNA haplotypes influence lifespan primarily by tweaking mitochondrial energy metabolism, OX-PHOS, and the resulting ROS production. That core link seems universal. Specific variants alter parts of the electron transport chain, affecting energy output and oxidative stress, which then impacts aging.
Okay, mechanism one, energy and ROS. What else?
Second, the absolute critical role of mitonuclear interactions. This isn't just a minor detail. It's a central theme everywhere we look. The effect of an mtDNA haplotype isn't fixed. It depends heavily on the nuclear genome it's paired with. Nuclear genes can buffer bad mtDNA effects, or they can make them worse. That dialogue is key.
Makes sense. And the other modifiers?
All right. Third, we see these effects are often modulated. Sex matters. The mother's curse shows how inheritance patterns create different pressures. And environment matters. Diet, temperature, stress. These can all influence mitochondrial function and change how a given mtDNA haplotype actually plays out in terms of health and lifespan.
So, putting all this knowledge together, it feels like it could have real implications down the line. How might understanding our mitochondrial blueprints change how we think about aging or health interventions?
That's really the exciting frontier. If we can definitively pinpoint mtDNA variants that make mitochondria more resilient, more efficient, or less prone to producing damaging ROS, could we perhaps develop strategies to mimic those benefits? Maybe nutritional approaches or even targeted therapies. And understanding those mitonuclear interactions better could be huge. Maybe future therapies won't just target one genome, but aim to optimize the communication between the nucleus and mitochondria to restore function. What's fascinating evolutionarily is this consistent emphasis on mitochondrial quality. It suggests that having a robust energy system isn't just about day-to-day survival. It seems deeply linked to the actual pace of aging itself. And understanding that might give us clues about how to influence that fundamental biological clock.
Think about this. The very essence of our cellular energy, passed down through generations from our mothers, might hold some of the deepest secrets to how long and how well we live. That wraps up this deep dive. Make sure to like the video and subscribe to support the SAN Science Experience. We encourage you to continue your own deep dives into the incredible world of science around you.